Okay, so welcome. Hi, I'm Julia Cooper, and I'm managing this project on early indicators of soil health with the Organic Research Center team and working with SRUC as well. So we've got a pretty jam-packed program today. Thanks for everyone who's joined. We're going to be giving you lots of opportunity to interact and provide your opinions. And we're aiming to run for about 2 hours, but we will give you a break. So I'm going to start just by handing to Philippa, my colleague, who's going to give you a little bit of information on how this workshop's going to run.
There we go. Hello. As I said, as Julia just said, I'm Philippa and I work alongside Julia and Catriona at ORC. I deal with a lot of the comms and the brand management, and I also help facilitate certain events like this. So just going to run through just a bit of sort of like housekeeping, and as it says here, just some session rules of engagement. It's going to be 2 hours, so it's going to be quite jam-packed. So to try and aid the smooth running of this, we're trying to, if you have any questions, if you would like to ask questions throughout the 2 hours, please can you put them in the chat? The hosts and myself, we will be reviewing those. And if we do get a chance to answer those, or if the hosts think that it's appropriate to actually respond to that in line with the discussion, they will address that at the time. There won't be any raised hands or anything during the discussions or anything, and we won't be able to address those again due to just to stopping and starting and dealing with the time limitations. So again, if you do have any questions, if you can put them in the chat and we will do that. As Julia said, there will be some breaks. There will be a comfort break, so we are going to keep you as comfortable as we can throughout the entire journey of this. The other thing as well is just to make you aware, all of your responses and everything and all your participation will be anonymous as per the participation information that you received. As you will see, that some of this is recorded. Some presentations done by Julia, Paul, Catriona, and Fiona will be recorded and be promoted after this, but none of your thoughts, none of your feedback will be. That will all be kept anonymous. So if there is any concerns about sections of this being recorded, by all means, either email me or email Julia, and we'll be happy to deal with any of your concerns. But it's just to address that. If there are any other questions about what I've just said, please feel free to put them in the chat, and otherwise, have a great session.
Thanks, Philippa. Yeah, so that was a great introduction. And the who's who bit of it, I think we've started already. So that was Philippa Hall from Organic Research Center, who looks after all our comms and events. I'm Julia Cooper, as I said. I also want to just introduce Catriona Willoughby. Can you give a wave, Catriona? You'll see who works with us at Organic Research Center, and you'll hear more from her later. And then we've got Fiona Fraser waving and Paul Hargreaves, both from Scottish Rural University College, SRUC, who you're going to hear from shortly as well. So that we're the team delivering this project on early indicators of soil health. So just going to run you through the program quickly so you know what to expect. So as I said, we've invited you here to learn more about what we've been doing in the project and just to talk about indicators of soil health. We know you've all got a lot of experience and expertise in that. And I should say, congratulations for being here because we did do a bit of screening. We really wanted people that were actively using soil health indicators in the field joining this workshop. So we're going to give you lots of opportunity to interact and give us your thoughts and opinions on what we've been doing. So we're going to start with a setting the scene that Paul and Fiona are going to do in a minute. And we're going to use this Menti tool. Hopefully it will work. It's one of these online survey tools that will share a link with you in a minute. So we can get some input from you at several times throughout the event. And then you'll see we're going to be looking at criteria for soil health indicators. So how we've been making decisions as we go through these different indicators that we're reviewing and selecting and give you a breakout room as well as another Menti on that. Then we're going to get into the details on the evidence for indicators of soil health, what we've learned so far, particularly these early indicators that Fiona and Paul are going to explain a little more about in a minute. And then we'll look at some more at the end rankings and next steps. So we're working towards narrowing down the various indicators we've been discussing in this project to a final three that there's going to be some further developmental work done on. So that's kind of the program, and there will be a break in the middle of that. So I'll hand it to Paul and Fiona now to talk a little bit of context and background. So take it away.
Thank you, Julia. My name is Paul Hargreaves. I work at SRUC, as Julia said. I'm part of the Dairy Research Centre, but my background is really in soil, and my colleague Fiona Fraser is based up in Aberdeen, working in the departments up there. We're more of a soils and plant focus than any livestock. I don't know if you want to just introduce yourself, Fiona.
Hi, I'm Fiona Fraser. I'm a soil biologist mostly. So I spend part of my time teaching and part of my time doing research and working with farmers.
And we were just here to really set the scene about the soil health card that hopefully people are using already. Just a bit about soil health to start off with. It's been seen as a key to crop production. To keep your soil healthy is to gain sustainability in that production. And it's a concept really that started about 10, 15 years ago to become more popular, where we were thinking about the soil in the whole, not just the chemistry of the soil that you're adding through fertilizers, but starting to think about the soil physics and the soil biology and how all this fits together. So you don't just concentrate on one aspect of the soil, but you look at the soil in the round really and try to improve or sustain the soil health that you've got through looking at the three areas of soil chemistry, soil physics, and soil biology. There's different aspects of this, and the scorecard that was developed for AHDB, I was involved in that development in 2018, 2019 to create this scorecard, hopefully reflects the three areas of interest in soil health. So there's the exchangeable or extractable chemistry part of the scorecard, including soil pH, the soil organic matter, which is more of the biological side of it, the VEST score, which is the visual evaluation of soil structure, which is digging a block of soil out to assess the physical types of the soil, so the soil structure and any limiting layer there that might be causing problems. And also earthworms is another kind of biological factor within the soil health. And this has been promoted now for the last 5-6 years through the Great Soils pages of AHDB and using the methodology there, and there's videos and information to back that up, including bands for assessing whether the outcome of your assessment for the different criteria are lacking somewhat and needing more intervention, or just coming out in red in the traffic light system, or amber if it needs just reviewing, that there's a problem maybe developing, or green if it's just continued monitoring to make sure that that there's no changes in that in subsequent years. And the times lagged for this was about every 3 to 5 years to do the soil monitoring and create a new soil card scorecard, which it was felt by HDB that with management interventions especially, that there may be a need for something that was more rapid than that showed changes on a more frequent basis, and what would be needed to be able to do that. Currently, it's soil pH, extractable P and K, Mg, soil organic matter, soil structure, and earthworm counts, where there's optional suggestions of potentially mineralizable nitrogen and the CO2 C burst methodology that's been suggested. but they're not critical to the soil scorecard at the moment, but maybe will be being discussed later on this morning. So basically, we was really trying to find something that didn't lag to 3 to 5 years, but was more immediate, to reflected changes, more immediate changes in the scorecard. So basically, what we're looking for today, and wanting your information on or opinions on, is the need for early indicators of soil health, things that can show a change within a year or 2 years. So this early indicator of soil health projects is funded by AHDB in response to lever-payers' interest. So there's been some response that more rapid scorecard criteria is needed to be able to show that these changes are happening, either positive or negative. So then some management can be done, or showing a trend in that direction to make decisions on how to improve or maintain your soil health. And it's also answering the question, is a recent change in soil management positively affecting soil health? So you don't have to wait 5 years to see if that management change has actually done something positive for the soil health. So basically, we're trying to look in the direction of change in a 1 to 2 year response. And I think this is where we come to the first set of responses from yourself.
So this is it's a program called Mentimeter, and if you can use your phone to scan the QR code, you can run through it on your phone. But I think we can also share the link in the chat, Julia.
Yeah, let me switch my screen now. Let's see if I can do this to the actual Menti site, and then you'll see the code. And let me just have a looky here. I've got multiple screens open. Here we are. No, that's it, right? Yeah. Yeah. So let me just copy that. I believe, oh, what's happening here? Yeah. So let me just copy that. Yeah. So I can put that in the chat. So you can join. Yeah. Let me put this in the chat here. Now
so
what's going on?
If you go to menti.com, and then it'll ask you to input this code up at the top of the screen.
Yeah. So there's a few ways to join. You can go to menti.com and input the code. You can click the link I've just put in the chat, which I think should take you right there. Or you could use the QR code that was on the previous one. Now let me have a look. Oh, we've already got, wow, okay. We've got quite a lot of people joined. I've got 14 so far, 15. How many do we have in this meeting? Can we see that somewhere? I feel like there should be a number of how many participants we've got here. Let me click my little participants. Oh, there we go.
23 in total.
23, and there's 5 of us. So we're looking for 18. Yeah, we're at 17 already. Let's see. Oh, 18. So I think we've got everyone. Excellent. Okay. So, and I guess you're all experienced with doing these things then. So yeah, we're here this is the beginning of just a little warm-up quiz, really. Oh, I think you were supposed to be running this one, aren't you, Fiona? I'll click and you can narrate the outcomes. Okay.
If you could move it along. Okay. So basically, it's just a yes or no answer to the first one. So do you currently conduct soil health testing on your farm or through your businesses? So if you could all just click either yes or no and submit, and then the results should refresh on the screen.
Are you seeing that in real time, Fiona?
I'm not sure. I can see the graph that's on the screen.
Can you see the little number bottom right? 14 out of 19 have responded.
Yep. Okay.
Yeah, 15.
So 15.
Yeah. Most people have.
I'm not going to respond, so I'm probably not helping here.
Yeah, well, we shouldn't really have joined, I guess. Not sure if any of us joined. Yeah.
Okay. So I think we can probably move on.
Yeah.
So
not surprising.
Which is good.
Mm-hmm. So we thought we were aiming to get people that did a lot of soil health testing on this, and we have, so that's good.
And then what we'd like you to tell us is which tests you use most frequently. So I think this is, again, it's just a click the ones that you use. I can't actually see the questions, Julie.
I think it's a free text. I think it's a word cloud.
Yeah.
It's possibly a word cloud or a free text. I can't remember. Let's see what pops up. Getting some good.
Yeah, just type in what you use and submit it, and it should all pop up on the screen. And the bigger the text is, the more people are writing that particular text. So some detail on chemical might be interesting if you're willing to provide it.
Mm. Vest is very popular, isn't it?
Paul will be pleased.
Love a vest, eh? And earthworms.
Okay, so we've got 10 responses, or 10 people have responded so far. So if anyone else has anything to add, that would be great.
Really interesting. Yeah. Above ground diversity. That's interesting. Yeah, plants as indicators. Yeah.
Okay, we'll just give you 30 seconds more and then we might move on to the next question. We're getting a good diversity, which is nice.
Yeah, really interesting. Soil cell. Yeah, we're looking forward to the breakout rooms where people can give us a little more detail on some of these
Absolutely.
methods. Yeah. Yeah. Worm counts, everything. Yeah. Great.
Okay, I think that will probably continue to
Yeah.
update. So if we could, well, wow,
Yeah.
if we could maybe move on to the next one. Okay, so
yeah.
a few words to describe what you find useful about particular soil tests. So is it about how easy they are to conduct or their availability commercially, or, you know, what else affects your decision to use a particular test? It should work in a similar way.
Mm-hmm. Yeah, I think this one isn't a word cloud. I think this is
No, it's
just,
a list.
yeah.
Uses the spade. Excellent answer.
I can see someone's asked about sharing the Menti results. I mean, we definitely will be including these in our final report. Yeah, that's right. Philip has just responded. Thanks, Philippa. Yeah, so I mean, this is one way that we're collecting data in this project really is through these responses that you're providing. So useful. Yeah. Oh, I see it's getting, yeah, I wonder if I can make that smaller. No, can't.
Okay.
Great. 11. So
All looking good.
yeah, so is everyone, oops, a few more coming in. Has everyone had a chance to put in their thoughts? We don't want to, I think that's the last Menti for this part of the program. But yeah, really interesting. And again, you will have, if you've not haven't had a chance or you think of something after we move on, you'll have a chance in breakout rooms as well to provide input. So don't worry if you suddenly think, oh, wish I'd put that comment in. Okay, so yeah, going back, should we move back to the rest of the presentation now, Fiona? Yeah.
Yeah, thanks everybody for participating. It's really useful.
Yeah, really useful. Yeah. So on to the next part. So yeah, in this project, we've been obviously judging early indicators of soil health based on certain criteria. And Catriona is going to talk to us now a little more about those criteria because we're interested to hear from you sort of what you think the right criteria are. So do you want to go ahead, Catriona? I guess I'll advance the slides for you.
Yeah, absolutely. Thank you, Julia. So hi, everyone. I'm Catriona. I'm a soil science and agronomy researcher at the Organic Research Centre. And a really central part of this project was trying to work out amongst ourselves and with help from people who were happy to input what the correct criteria were for assessing the early indicators for soil health. So if I could have the next slide, please, Julia. So we were helpfully provided originally by AHDB with some criteria against which we should assess each of our indicators. And they made quite a lot of sense initially. So of course, any indicator that we chose, there must be really clear links between the performance of that indicator and soil health itself. There must have a good backbone of robust UK applicable data so we can have confidence that it's relevant in a UK setting. they need to be useful to land managers, the people who are actually managing the soil and conducting the tests and making those decisions. Because they're an early indicator for soil health change, it's obviously really helpful if they're actually quite sensitive and responsive to management change, although sensitivity can be a bit of a double-edged sword because you don't want them to be so sensitive that they'll be really different regardless of when or where you test them. We also had considerations for the actual analytical costs, and this tied a bit into the commercial availability and the potential for commercial availability of some of the indicators that we were considering as well. And then finally, we needed to look into the spatial variability, which ties into indicator sensitivity, but also in terms of how many samples would you need to collect to be really confident that that result that you have is representative of your managed area. Can I have the next slide, please, Julia? We also wanted to assess the early indicators for soil health against the provisioning of various different ecosystem goods and services. When we think about soil health, I certainly tend to think about it on a landscape level, supporting crops, supporting our natural spaces, supporting biodiversity and livestock. But obviously a lot of these interactions in soil are taking place on a really very small scale. And what we wanted to do was assess the ability of each of our metrics to support food and fibre production, nutrient cycling. We differentiated out between nitrogen and then phosphorus and potassium. In the capacity of the soil to support water regulation, either to improve resilience against flooding or improve resilience against droughts, and climate change mitigation. Oh, thanks, Julia. And then we also had to consider the context within which the indicators were useful. So land use was quite a key consideration for us. We wanted to make sure that we were selecting indicators that were appropriate in an arable and in a grassland context as well. And I think I have one more bit, thank you. And then we also wanted to make sure that the indicators that we were selecting were also applicable across different soil types and soil characteristics as well. And that is quite a large list really of considerations that we had to make. So I think I've summarized them all in a little table on the next slide. So these are, I've separated them out into little categories here. So we had our kind of 6 components of the AHDB criteria for consideration. We then also added in our ecosystem goods and services as well. And then we looked at the context of each indicator, and we differentiated these with quite a broad brush initially. So we looked at whether or not they were applicable arable or grassland, and also in terms of soil texture, because there's a really good background understanding of soil texture in the UK as a rule. So these are our current criteria. I believe we're about to move into a breakout room to give everybody a chance to discuss the different aspects of criteria, what's a priority for you. Julia and Fiona have set up some Padlets, I believe, so that we can aid those discussions. So I might hand back to you, Julia, just to be in the driver's seat for this bit.
Yeah. Okay, so it's actually, this is my part now. So I'm just going to talk a little bit about the actual indicators of soil health that we've been reviewing in this project, which is probably what you came along to learn more about, and we've kind of kept you in suspense till now. Of course, there are dozens and dozens of different indicators and methods and protocols for assessing soil health. Some of them Paul and Fiona introduced to you earlier that are already in the AHDB soil health scorecard. For this project, as we've explained, we were tasked with looking at what we call early indicators of soil health. So indicators that might show up an effect within the first, within a year or two of a change in management on your land. So at the proposal stage, we were asked to already produce a list of 10 that we would be looking at initially. And then we've been going through this sort of process of narrowing them down. So you can see there the 10 that we proposed to AHDB at that stage. And I'm just going to run through them quickly and a few comments on them. I see something in the chat, but I'm not going to look at that. Hopefully Philippa can respond to that. So these are the 10.
What is PLFA?
I'm going to explain. Yeah, okay. I am going to explain that. I'm going to go through each one just quickly. Oh, I've lost my pictures of people. Okay. Yeah. So yeah, I'm going to go through each one. So basically, but I also want to explain how we've been doing this sort of sifting process. So what we first did was take that list of 10 and narrow it down to our aim was 4 to 5 indicators that we were going to do a more in-depth review of. So we used what's called a Delphi process, where you get a bunch of experts together, and we had about 10 or so people that we put into our expert pool, including people from industry, commercial people, agronomists, and scientists, and we went through these sort of in an iterative process to narrow it down, involving some surveys and workshops, etc. So we were building, a consensus basically, and I'll explain to you how we did that. But just to go through the different ones very quickly, and these are some comments that came from the experts as we reviewed them. I won't say which ones we've shortlisted yet. At the end of my section, I will. So PMN, potentially mineralizable nitrogen. I'm guessing most of you are familiar with that. And it's some concerns about it. Currently, thresholds are quite broad. It does represent an easily degradable pool. It isn't maybe really linked to any functions or thresholds. That was one comment anyway. So some concerns about it, but also there was some interest in it. It was one that was identified. All of those 10 that I showed on the previous slide were identified as being what we thought would be fairly rapid in responding. Active carbon, some of you might have heard of that one. That's offered by some commercial labs. POC is a short form for the particular method that we decided to focus on, permanganate oxidizable carbon, which is offered by some commercial labs in the UK. It was expected it would be highly correlated with PMN. There's a concern of a lack of interpretation frameworks, but it is likely an early indicator. So that's another one of those 10 I've shown you. Crop rooting, an interesting one that there's some protocols being developed by our colleagues here at ADAS in a European project on using crop rooting. There's with some direct links to functions. Obviously, your crop itself is an interesting indicator of what the soil is like below the ground and how well it roots. Farmers could actually implement it themselves, although there might be some training required. So that was another one of interest. PLFA, yeah. So someone asked about this. Phospholipid fatty acids. So it's a method to characterize soil microbial communities. So it's a laboratory method. You can get groups of bacteria and fungi from doing that analysis. So sort of a precursor to the more of the DNA-based methods currently being used. You can use it to derive fungal to bacterial ratios, which we know there's a lot of interest in the industry in. But then we had within our expert advisory group some commercial labs working on developing PLFA, and they actually said they didn't think those methods were ready for deployment yet. And also there's a general uncertainty over how you actually interpret a fungal to bacterial ratio. Although you can use PLFA for more than that, of course, but that is one use of it. So that's another one. We looked at soil food web. I discussed that. There was concern there was a bit of a lack of evidence on the value of that, and it may not be so sensitive, but it is popular with farmers, and some farmers are doing those analyses themselves, extracting fungi and bacteria, et cetera, and looking through microscopes. So it's really engaged farmers in soil health and soil biology. So that's another one. Aggregate stability, a good old-fashioned measure of, just as it says, how stable are those soil aggregates? How resistant are they to erosion and breakdown? Thought to be particularly linked to water regulation. But there's a picture of one of my students a few years ago doing those measurements in a lab using this wet sieving apparatus. And so it's quite time consuming to do and labor intensive. So that's a bit of a drawback to it that needs to be taken into account. So there aren't yet commercial sort of rapid methods that commercial labs are offering for aggregate stability. What else have we got here? Enzyme assays, we looked at those as well. So you can see at the bottom there of this image that the FAO has released some protocols recently on how to measure for those various enzymes listed there. And they all have different functions in soil, doing different things within the sort of process of degradation of organic matter and release of nutrients, soil respiration. So all indicators of biological activity and maybe more specific functions. But again, questions and concerns about how you interpret them. But some labs are developing these protocols for commercial release. So that was when we discussed CO2 bursts. We discussed, again, available commercially, but thought to be linked quite closely to active carbon and also potentially mineralizable nitrogen. So that's just a picture of how it's done in a lab. You know, it's that you take an air dry soil, add some water to it in a sealed bottle, measure how much carbon dioxide is released in 24 hours. So it just gives you an indication of sort of available carbon and potential of the microbial community to metabolize that carbon. Sorry, I'm rushing through these quickly. If you want clarifications, maybe put them in the chat and I'll look at that in a minute. And then earthworms. We did include earthworms to have another look at them, although there's a concern that they're what we call a lagging indicator, so something that takes quite a while actually to respond to management change. And we're looking for what are known as leading indicators, indicators that will change quite quickly. So these are just some of the concerns raised and points discussed with our experts. Let's see. Yeah, so then we did what's called a Delphi process. As I've already said, it involves consulting with experts, running some surveys, and then we had some final workshops. Ultimately, we had a vote. So we were trying to reach a consensus. Consensus you normally think means 100% agreement on something, but in a Delphi process, you don't go for that. You set a threshold. So we set this idea of 70% agreement. So we then what did we do? Oh yeah, we first of all narrowed those 10 down to 7 during a workshop where we identified the ones we thought were the no-brainers, and then we identified a couple of what we called dark horses, so ones we thought might be worth looking at in more detail. And then we asked our experts to vote. So we were looking for more than 70% agreed to include that indicator in our sort of next sift, let's say. So those are the 4 that we ended up that most of our experts felt were of most interest to include. So PMN, crop brooding, active carbon, that's that POC, and aggregate stability. So the experts were asked to vote for each one, include, exclude, or not sure. So the only one where we had a consensus to exclude was soil food web. So 75% of the experts felt shouldn't include that one. And then we had these sort of not sure ones, PLFA and enzyme assays, ones we might want to, at some point, maybe need some more research and exploration. But so that's where we got with the experts, and that's how we ended up with these 4, which Catriona is going to talk about now, and how we've now been sort of reviewing evidence and synthesizing evidence on that. But just let me check for a minute to just double check there's no really burning questions on what I've just said. Oh, there's a few in the chat. Yeah, so let me just check here. Oh yeah, the PLFA, yeah, so that's fine. So I think we can go on if everyone's happy to Catriona's part. Is that okay? Yeah, so we'll go on to that.
Yeah, so I'll take over from this point. So Julia guided a lot of the expert panel and Delphi process. And then once we had our shortlisted 4 indicators / methods there, we then had quite a lot of information to review from various different sources. So I'm just going to quite quickly talk you guys through the approach that we did use and our initial preliminary findings from that. So we knew that we had these 4 indicators that were to be taken to the next stage. So we carried out an evidence review looking at key academic literature, statutory reports as well. There's an awful lot of work being done to identify a healthy soil indicator for England and Wales, for example, where they also reviewed a lot of evidence and used expert panels. So we were able to utilize some of those reports as well. And we also carried out consultation with some of the experts that we had available to us, partly because, particularly for crop rooting, that's an indicator that has been explored somewhat in the academic literature, but It's not particularly commercially done yet in the UK. So we were able to use some of the experts that we had access to gather a bit more information on that. But in order to get rankings out of those quite distinct pools of information to start with, we used a method which is called a logical sieve. So I'm going to show what that looks like. It's a big Excel spreadsheet, basically. And Julie, if we could just go to the next slide. I think I have blown it up slightly and just gone over the outline just so that it's a little bit more understandable what this process actually was. So at the end of the day, it's an evidence review. We had our criteria for consideration, which you'll all now be very familiar with at the top of this list. So along the top, we were looking at the sensitivity to management change, analytical cost, spatial variability. We had a module that was looking at ecosystem, goods and services, applicability across the different land use types that we were considering. And soil texture as well. By taking this approach, we could quite systematically review the information that we had available and look at scores for each of our criteria. We could differentiate according to our sort of 6 AHDB criteria and our ecosystem goods and services provisioning. And also look at and also make sure that we were considering land use applicability and soil texture. And then down the bottom, we had a look at indicators as well, and then the scores themselves, which were derived from a different metric for each criteria, which sits within the sieve as well. The eagle-eyed among you may have noticed that we've got an expert group weighting factor, which we'd considered earlier on in this workshop today. The information that we gather during the course of our industry workshops will also go into determining weighting factors. So the feedback that we gather from these workshops will certainly inform our results and our final reporting as well. So we've taken all that information, basically, we've turned it into a rather huge table, and then it all condenses down incredibly neatly into our preliminary ranks, which I believe are on the next slide, Julia. Oh yeah, we had another slide here about potential mineralizable nitrogen. I think that might have gotten a bit lost, sorry, when we did it. So it's an optional method, but yeah, we can go into that. So,
oh, sorry, do you want me to skip that one?
Yeah, that's okay.
Yeah, okay.
That's my bad. So basically, we had our 4 indicators here, and we've ranked them based. I've just summarised the scores across the 6 AHDB criteria and the ecosystem goods and services as well. The current rank's colour-coded. So number 1 at the moment in red is aggregate stability, and this was largely based on it had a very high score in terms of provisioning of ecosystem goods and services for this indicator. It didn't do as well in our sensitivity cost sampling criteria. And then we had active carbon, which again was kind of our one of our best scorers for the provisioning of ecosystem goods and services. And then the highest ranked criteria for the HDB was actually crop root architecture, which I think reflects it's quite a practical test to carry out. There's some training available commercially in the UK. It ties in quite well with some of the other indicators which are already utilised in the scorecard. And then, so that was ranked 3rd overall currently. And then we've got potentially mineralisable nitrogen coming in at the moment in 4th in terms of our preliminary rankings. And this is based on the weightings with the expert group, but we're going to go back and have a look at that again. So yeah, those are our preliminary results so far. And yeah, Julia, I think we've possibly got another opinion gathering exercise after this point, now that everyone's seen the ranks that we came up with.
Yeah, so next steps. So yeah, I think we're coming just to the end. We've got 5 minutes left, which is perfect. Yeah, what are we going to do next? So we're hoping to take 3 indicators ahead to develop some kind of interpretation framework. So this input you provide is really important. The ranking we showed you isn't necessarily the final ranking showing the 3 we're going to take forward. So we're going to take these comments into account. We're running a similar workshop this afternoon. And I think, really useful to get your input, especially these comments like you've been making, Abby, about the real expectation that these were going to be things you could do yourselves out in the field, which wasn't, we weren't given that those boundaries really. We didn't exclude in-field measurements or lab measurements. We just were looking at everything really. So that's really quite useful. And that's interesting. Now John's just pasted something in there. Yeah, that's good. That's useful as well. Yeah, so we'll be looking at the results of these two workshops and then choosing three that we're going to take forward. Looking at then working with AHDB on how we can integrate those into a new improved scorecard that includes some more early indicators of soil health. And we're also, knowledge gaps. So there's always, with projects like this, there's always a part where you talk about what's the next sort of research that needs to be done, what new projects need to be done. And so we're going to be looking also at some areas for future development. So that's sort of what we're going to be doing next. Yeah. And good point there too, Tony. Thanks for that. So that's everything I had to say, really. Yes, it is, Matt. The afternoon workshop's identical. Yeah. And numbers, I think we just wanted to give people a bit of an opportunity to if they couldn't make a morning, to go to the afternoon. I think some people registered for both possibly, but I think Philip has sort of screened those out. So no, you're not you won't miss it. You're not expected to come this afternoon as well, because it would be very repetitive. Yeah. So does anyone have anything they want to add at the end? Any final comments? I mean, I think I just want to thank all of you. Really, really useful input. Thanks for engaging with all these interactive activities. Really, really valuable. So thanks a lot. And thanks, of course, to AHDB who are funding this work and genuinely trying to respond to their levy payers' needs and interests. Okay.